So let me welcome everybody to our Monday seminar series at the HNRCA at Tufts. It's really my privilege to introduce today's seminar speaker, Dr. Mario Faroozi. And judging from the chat box right now, Mario is well known to many of you. For those of you who do not know Dr. Feruzzi, he serves as the brand new director of the Arkansas Children's Nutrition Center, which is one of the 6 human nutrition research centers within the USDA ARS, which of course includes the HNRCA. Dr. Feruzzi obtained his doctoral degree in food science and nutrition at Ohio State University, after which he has held positions at Nestle, Purdue University, and North Carolina State University prior to moving to the University of Arkansas for Medical Sciences. Mario is very active in multiple professional societies such as the Institute of Food Technologists and the American Society of Nutrition. He currently serves on multiple advisory boards, including the USDA National Agriculture Research, Extension, Education and Economics Board, the International Food Information Council, as well as leadership roles at IFANS, formerly known as ILSI North America. Dr. Feruzzi's research focus is on key issues of how the food matrix and food processing impacts micronutrient and phytochemical stability, bioavailability, and metabolism. He has published almost 200 peer-reviewed papers and is also the co-editor of 3 textbooks. He is exceptionally well-funded with extramural grants received from the USDA, NIH, USAID, and Bill and Melinda Gates Foundation, among others. So without further ado, I would like to hand over the virtual podium to Dr. Mario Faroozi to discuss the variation of micronutrient and phytochemical bioavailability in plant germplasm collections in order to improve delivery and nutritional impact. Take it away, Mario, and thank you very much.
Thank you, Sarah. Just wanted to thank Sarah and folks at the Human Nutrition Research Center on Aging at Tufts for giving me this opportunity. As a new center director myself, it's always great to learn more and more about what all the other centers do and to get a chance to interact. So I really cherish this opportunity, and like I said, hopefully it'll cut into areas that are familiar to some of you on the side there at Tufts. So yeah, today is a little bit different. I'll give a lecture on some work that started, I would say, and see if I can get this to advance, started actually while I was at Purdue, both in food science and nutrition science at Purdue. Really grew from my focus on food matrix when I went to NC State to join the Plants for Human Health Institute, where I started turning more and more back to the field and the agronomic side to try to understand micronutrient phytochemical content in germplasm collection, help breeders, but also to learn how to translate what was happening in the laboratory to the field, and then from the field to the table. Taking that full circle, it give you a little bit of an idea of what hopefully I'll bring to Arkansas Children's in terms of connecting the dots back to agriculture, and the USDA connection there for me is quite relevant in terms of how we think about nutritional impact. So when I think about it, I started years ago thinking from the concept of cell or seed to cell, right? So, and then back again. So what we learn as we learn about plant genetics, as we learn about agronomic effects and post-harvest management as it relates to micronutrients, which is my area of interest, primarily provitamin A carotenoids, but also bioactives such as phenolics. What we learn about that in the agronomic space is typically work of agronomy, like worked with a lot of plant breeders and geneticists and in this space. And then of course, us as food scientists are working in the area of post-harvest processing, food formulation, and then getting it to the point of a consumer actually eating it. And I was always told when I started at Purdue, coming from industry and then going to academia, well, once it touches the lips, it's no longer the domain of food scientists. Now you're talking about absorption of nutrients and eventual impact. That's the domain of the nutrition scientists. this segmentation with occasional touch points is what always kind of drew me to the space of food. And so thinking about how we actually bring this all back full circle was really the driving point of a lot of the research that I was able to engage in with really great collaborators. And to give you an idea that it isn't a linear process, we don't start just by breeding and then measure everything sequentially. I think we now know that this process is circular. We also know that there are touch points of things that we would not perhaps have predicted. The fact that plant genetics might impact not just agronomic traits, but also stability through post-harvest management of certain, let's say, labile micronutrients. They also impact how relevant it is for processing. And might it impact digestibility? Absolutely. This is a key area of research, for example, in cereal grains and protein digestibility. But we also now are learning more and more on the nutrition side about host genetics and how it impacts response to food and diet, and also digestibility and absorption of nutrients and interaction. We could also put into here the microbial profiles and microbial genetics. But what we do know now is that arrows point in every direction. And the key thing here is to try to bring back information to pertinent points that can help us improve that circle and improve ultimately the impact of what we see. So for today, what I was going to do is give you a little bit of the work that we have accomplished and are now currently taking to another level, I would say. And I'll start really with polyphenols, which is one good example I'll give today. I'll also go into carotenoids and some of the work that really drove us into this area on the provitamin A side through biofortification. But let's start with polyphenols, which have been associated with many health benefits. And, Tufts as a center was central in this story. Blueberries and grapes and other wonderful research that came out of the Tufts area as it related to aging, primarily because these polyphenols in fruits, vegetables, coffee, tea, and other botanicals have been shown to have the ability to modulate many of these processes of oxidative and inflammatory stress, altering nutrient digestibility and absorption that could impact blood lipid profiles. And now we're talking about the opportunity to impact disease risks and outcomes, especially cardiovascular, cognitive, again, which is Tufts has certainly played a leading role in, cancer, bone health, obesity, and diabetes. And some of the really key things that have come out of this more recently, of course, are this area of gut microbiota response in and of itself, and how these polyphenols may in fact serve very much in a prebiotic nature. So they become very good targets of study. And then the question becomes, how are these compounds delivering their benefits? And then how are they going to be delivered to consumers in terms of foods or dietary supplements? So when we think about polyphenols, we think about them traditionally in the context of food, where they're interacting with carbohydrates, fibers, proteins, lipids, and other phytochemicals. Post-consumptions, these can be digested, but then there's also interaction with the microbiota and the ability to alter the microbiota composition and functionality. And we think about these factors as driving what I would say the health effects, especially in the context of how they interact both with the microbiota and can be converted to biologically active metabolites, these postbiotics. And it becomes a combination of it's impacting the bacteria, but it's also bacteria metabolizing polyphenols. And the combination of these metabolites and shifts in the microbiological composition of the gut could in fact be driving many of these health effects. So it is not a simple story. And the question then becomes one of how, is it just about polyphenols? Is it just about postbiotics? Or what about all these other components and how they alter this chain of events, if you will, in the gut? And this kind of came to a head to us very early on. This was about 2014, 2015, where we were working with Connie Weaver at Purdue at the time before I left to go to NC State, and then working with Teresita Belito, who was at IU School of Medicine at the time. She actually is now at University of Arkansas for Medical Sciences. So we're working together again on, this was a very large NIH R01, which really combined more almost like a collaborative R01. It was a very significantly larger than a typical R01 grant. The notion of blueberries and berries in general, I say, but blueberries and bone. And of course, I think most of you are aware Dr. Weaver's interest in bone, but the connecting point here was looking at the polyphenols in blueberries and getting an understanding of their specific metabolite profiles that were interacting with the NRF2 pathway that could alter the antioxidant and inflammatory response to then have effects on bone. And so we had condensed this project, which was supposed to be a program project, down to one project and plus more, because that's why it says berries, but our focus became on blueberries. And if you could imagine, those first few meetings got to be kind of interesting, because here I am coming from the food and ag side, and we said, well, what do you mean by blueberry? What are you interested in terms of blueberry? And at the time, you know, just thinking about what a consumer might be interested in picking up at the store, the difference in what they pick up in Boston versus Charlotte, North Carolina versus West Lafayette, Indiana, could be vastly different depending on the time of year. And luckily, working with Marianne Lila, who was a key collaborator in this project, we understood that there was a fairly significant variation across the vaccinium species. And in fact, what you're dealing with is thousands of individual vaccinium genotypes. And how do you select either the ones that are culturally or I would say more relevant economically and to the consumer versus ones that could be exciting for research? And luckily what we had is a set of data from Marianne's group in North Carolina that had already been looking at collecting this, what we would say phenotypic data of anthocyanin profiles over 267 genotypes, most of which were economically relevant either for breeding or for production. And so you start looking at it and say, well, what does this variation look like? If it's all pretty close, then we can pick any blueberry and it'll be fine. But the NIH was pushing us to say we need things that are also relevant and translatable to humans. And so we don't want you studying experimental varieties. So we did a simple PCA plot just to capture what that variation looks like. And you can see that's pretty well spread. In these cases, we were able to say, yeah, there's pretty uniform variation, if that means anything, to those of you. This is what we like to see, a broad spread, broad diversity of anthocyanin profiles across a couple of principal components. So this gave us an idea, well, how do we represent that if we are going to go into preclinical studies or clinical studies where you can't afford to study every relevant genotype? So we put together a strategy to actually select out about 5 to 6 genotypes that represented the broader diversity across the second principal component and some across the first. The rationale for that was the first was really all about total, while the second was more about qualitative differences in anthocyanin profile that were in these berries. And we were able to select out and then do an in vivo bioavailability experiment in rodents. And in doing this, basically acute study in rats, we were able to kind of select out and say, all right, well, if we have blueberries like Ira, Montgomery, and Onslow, as well as one of these crosses, how does it compare in terms of content? As you can see in blue, Montgomery was very low, Ira was very high. But when you looked at the blood levels, the irony was Montgomery was quite high, Ira and Onslow were quite low. And then here you have wild blueberry, which is a composite coming from the Wild Blueberry Association as a control. So the berry that had the lowest amount of anthocyanins had the highest blood response of the cumulative metabolite profile coming from anthocyanins. Now, at the time, we were looking primarily as anthocyanin glucuronides and main metabolites of the host. We weren't looking yet at the microbial metabolism, but this gave us an indication that you can't just go by, let's go with the one that has the highest. So spinning that forward a few years, just wanted to highlight Terry Bolito's work, where they were able to look in their NRF2 knockouts mice, the fact that in response to 3 blueberry genotypes, in particular, well, 2 genotypes in a composite, Montgomery and Ira, those 2, Montgomery that was quite high in bioavailability and Ira that was quite low but high in content, versus a wild blueberry kind of control for us, which we know is a pretty good composite of a low bush profile. But actually, out of all of these, only the Montgomery blueberry genotype was protective in OVX female mice for bone loss and increased body weight. Interesting thing, it was associated with endogenous antioxidant response, and alpha and beta diversity changes, which really highlighted the fact that, hey, we must have had a higher response from blueberry phenolics in the blood from Montgomery, which is what we had seen before. Make a long story short, I'm not a bone person, so I don't want to get into the mechanisms of all of this, but we were interested in, hey, is bioavailability really a predictor here? And is there diversity of response? And is this something we need to consider as we think about any other study we try to do? So how do we go about explaining why Montgomery is perhaps better? And that's not going to be the rest of my talk, but the thought is, how do we actually begin to understand this component in a way that's meaningful, not just for the biomedical side and the nutritional side, but also for, hey, how do you breed for better products? So we started thinking more and more about, hey, how do we get to understanding these areas? This is a part of what our research program has been now for 15, 16 years. And we study phenolics from food, how they're released through digestion, how they're absorbed, metabolized in the intestine through host metabolism, host metabolic processes, leading to metabolites that then are further metabolized in the liver, to further metabolites, host metabolites, distribution, and ultimately excretion. But the chemistries of what happens in the small intestine are a big focus of our area of research. And by that, I mean, we are very interested in studying food matrix effects. So just because something isn't as high in phenolics, there could be interactions and processes driven by the matrix that are helping it be more effective. We want to know what those are, so we might be able to breed those into berries that have higher phenolics, so we can actually become more and more effective at delivering benefits, perhaps from the fewer servings that consumers actually consume. So this includes interactions with micronutrients, macronutrients, et cetera, and form of the product, including dietary supplement forms as well. On top of that, we have had a growing interest in understanding how these phenolics are metabolized by the gut microbiota. And of course, the interactions with the food matrix there are critical. Are you putting it in with fermentable fiber? Fruit is pretty rich in fermentable pectins. So are there, in fact, benefits with certain structures of those fibers that we should be considering if we're trying to derive the best benefits of the phenolics? So these are all types of questions that we could explore for years on end. So we had to start somewhere. So where did we start? Well, in the context of this berry and bone project, we actually started by comparing what happens, the difference between some of our preclinical work and clinical work that Connie was going to do was going to be feeding berries or freeze-dried powder versus potentially extracts. So how do we rationalize if there's any difference or if there are toxicity concerns, which is something that the NIH was very interested in us doing. So as part of a 90-day toxicity study looking at berries and extracts, the question being, can you go toxic on an extract or can you ever go toxic on a whole food? And we were able to basically track down relevant levels from a diet that you would get from freeze-dried berries, 50 milligrams of per kilogram body weight, and then extract amount made from the same berries in increasing content. So 50, 250, 1,000 milligrams per kilogram body weight. So we really overloaded these animals to see if we could get ultimately some toxicity. But as part of that study, we were able to map out the main phenolic metabolites that were also being generated by the microbiota and then also by the host conjugates of those microbial metabolites. This includes chlorogenic acid and different flavonoids, anthocyanins, flavanols, flavonols that are present in the blueberry. Cinnamic acid, phenylpropionics, phenylacetics, benzoic and empiric acid, as well as benzaldehydes being the primary breakdown products that we see from the microbiota of these compounds. So tracking to see if there's a matrix or a dose effect on this. So just to show you on a heat plot, here's the fruit, and you have a 90-day study, and you can see by each class, berry phenolics, phase 2 metabolites, phenolic acids, which are microbial, and then phase 2 conjugates of those, and then ultimately hippuric acid. And what we find is over time, you see greater and greater, pretty good stable relay from food with a high excretion level of hippuric acids, representing 6 to 39% of that overall excretion. But when you get to extracts with a low level of extract, there's some similarity to what you see from food. There are a little bit of a subtle difference in the sense that we see higher levels of microbial metabolites, perhaps, and lower levels of butyric acids. But what's interesting is as you get to higher and higher doses, you begin to back up and see more and more in the urine of actually parent compounds and earlier stage metabolites. And so greater excretion levels actually accumulating in this phenolic acid range than anything else. So we do see differences in ultimately what's going to be in blood if you change the form and you change the dose. Again, this isn't surprising, but what's driving it, especially the form component, which is actually quite interesting to us. Interestingly enough, in the same study, Dennis Cladis, a student of mine and Connie's, actually was able to kind of but study changes in the gut microbial profiles, showing that, alpha diversity, blueberry, low dose, and medium could actually go up a bit. But once you get to the really high doses, you begin to disrupt that alpha diversity and drive up, for example, Bacteroides populations there. And you really see this in terms of the, the beta diversity, in the sense that the high dose group begins to behave quite differently. So again, just a point of us looking at how we're going to translate this, you begin to start seeing these differences in form. So back to what I was talking about before, where do plant genetics play a role in this? Well, this already tells us we can move the needle based on dose, but what are other factors that drive that effect? So what are other factors that we know from food, right? Solid liquid, all the macronutrient interactions, et cetera. These are from 15 years of our group studying phenolic bioavailability in animals and humans and in vitro models. But then if you take that all from food and you look at something as like a blueberry, well, there's a lot of factors there as well that actually play out. Skin thickness, size, fiber content, titratable acidity, sugar content, the actual polyphenol profile. All these things fit into those factors that I have listed on the side there in some way. And so at the end of the day, what is important about that? That's all controlled by the plant genetics. So we know we can alter those factors by the plant genetics. We also know that some of those factors will be altered by where it's grown and how it's grown. So the environment and the breeding world and genetics, what we call that G by E. As a food scientist, we also know that you can process this stuff into different products. So that's called G by P, or genetics by processing, or G by E by P. So this can go on obviously indefinitely, but I'll give some examples of all of these here today, that not just on phenolics, but on carotenoids as well. But the key thing is you can understand now that those matrix effects might very well be enhanced or created in a negative way, if you will, by plant genetics. And it could give us a way to learn more about how to work backwards, if you will, because all of these factors are going to serve to impact bioavailability, perhaps, and ultimately activity. So this gets me really to the project that we had developed when I was at NC State, along with Marianne Lila, Colin Kay, and Massimo Iorizzo. Massimo, fellow Italian, and also the plant geneticist that I worked with mostly here. And Colin, who many of you probably know from the phenolic metabolite side there. This was a project that was funded by the Foundation of Food and Ag Research, where it was really about closing the gap in delivery of fruit and vegetable benefits. And our model system was blueberry amongst one of them. We also looked at spinach, so I'll show data on that. But really the first aim is what I'll talk about today, which is breeding better fruits and vegetables, not for content, but also for bioavailability. So how do you do that? When you think about breeding, we think about breeding targets. And so when you think about that, you have to have something that you can phenotype. Otherwise, it takes too long. We can't do a bunch of animal studies to measure thousands of different blueberry genotypes. We can't do human studies to do that. So how do we emulate what the breeders do or what the geneticists do? And in this case, working with Massimo, we already had a little bit of a head start because we were looking at diversity of these phenolic metabolites as it related to fruit quality traits, really to try to establish a framework for genetic studies for polyphenols. So 128 blueberry accessions, 2 years. These were grown in Corvallis at the National Clonal Germplasm Repository. Samples were collected, sent to NC State. 30 health-related polyphenols were analyzed, over 30. These are the classes. You'll hear me talk mostly about classes because the data gets a little too granular. And then we looked at does this relate to fruit quality traits, skin thickness, titratable acidity, etc. And this is Massimo is part of his broader work in Vaccinium. So this fits in really nicely to give us some indication of how it was going to work. And so what did his group really do? They did a really wonderful job of doing what plant geneticists and breeders do. They can look at the ploidy level and understand if there's any correlation or proxy that they can use for any of these traits. So in this case, fruit size as a proxy of fruit weight. This is pretty common sense stuff, but then they could also look to see if there's any correlation of fruit size or weight or any other factor with phenolic content. And here they were able to find a negative correlation, which one would anticipate. Smaller the fruit, perhaps the less moisture there. I think this has to be tightened up on solids, but you begin to see, in fact, the ability to predict these things out and then select out certain accessions that have perhaps a higher content of anthocyanins, in this case, per size. So you can, if you're interested only in content, this is the type of data you would look at. Now, what's really neat about that, doing these types of studies, is you can also get into the factor of genetic control. And so looking at the tetraploid, which is primarily the ones that we would say are perhaps most interesting for us from a commercial perspective as well, this is a plot of broad sense heritability. So this is the variation in anthocyanin. These are individual anthocyanins, individual flavanols and flavonoids and phenolic acids as it relates to that is driven by the genetics. Okay, so getting rid of all the other variation, and anything over 40 is typically saying that there is moderate to high sense heritability. So this is a heritable trait. And you can see, for example, with anthocyanins, they all cross that line. Some of the flavonoids do, and some of the phenolic acids do. But the point being that there is some genetic basis for the expression and the content of these in the fruit. Again, things that you might think are already known, but need to be established before we look at bioavailability. Big component of that is then the study of, and Massimo's group is very interested in studying acylation, which is the structure here of a basic anthocyanin. We normally think of the modifications on this B ring as either hydroxyl or methyl groups, but acylation on the glycosides to other phenolic acids is also a key factor driving stability to heat, can enhance molecular interactions. It's been known to affect bioavailability, probably because you're putting more phenolics in the lower gut, okay? Because now you'll get differential metabolism by the gut microbiota. So this notion of acylation is important, and I'll show why here shortly. But do the genetics that drive that lead to altered physiological response? And so he had been, in his group, focused on understanding the genetics that drive acylation of these anthocyanins. And now we wanted to piggyback a little bit on his work to better understand, are there also genetic factors that can drive bioavailability? So that became the question. Does bioavailability of blueberry phenolics vary to the same extent as the phenolic profile, or is variation driven by other factors under genetic control or not? That was a question that we had right off the bat. Like I said, you can't do human studies, so we have to rely on an in vitro surrogate of this. And we had been working quite extensively over the years on just an upper GI model. Now we've added a lower GI model to this as well. I won't speak about that today, but the upper GI model is quite interesting because it simulates oral, gastric, small intestinal digestion and disintegration of the food. So food factors can in fact be measured that affect digestion, will affect bioaccessibility. And we can express that in relative terms. So the percent of the phenolics that are extracted, if you will, and made available for absorption, or in absolute terms, the relative bioaccessibility times the serving size. So you can actually get an idea of the milligrams available per serving of blueberry, for example. So we use this model over the years in our group to kind of correlate to a lot of our human work and animal work, understanding relationships from tea and cocoa and juice and wine to looking at polyphenol protein interactions. We've studied whole grains with this, and we've even looked at carotenoids and lipids. And I'll get into examples of carotenoids here in a second. But the point is, we've been using these types of models for lots of years. We have lots of good correlative data to human studies to know that they're predictive. but they are also cumbersome. And to point that out, when I first said, yeah, we could do this, I didn't stop and do the math. So when speaking with Massimo and others, they said, yeah, we want to do, several hundred of these genotypes. Well, you multiply that by replicates, and next thing you know, you're doing thousands of individual digestions. And the old way that we used to do this was 50 ml tubes by hand pipetting, limited by the number of spaces we could centrifuge to isolate bioaccessible fractions. To make a long story short, we could do about 24 to 30 samples a day. So if you got to do 2,000 to 3,000 digestions, that's a lot of days. So we had to quickly adapt to something that was a little bit more high throughput. So we adapted a robotic system to actually do this. And now we've taken everything down to microplate, 96 well collection plates, and high throughput methodology using UPLC, mass spec, that allow us now to get up to 200 to 300 samples a day. So now it's much more manageable. And the good news is low throughput and high throughput correlate quite well. We're able to look at over 30 phenolic species at the time when we did this study at once in blueberries to estimate bioaccessibility of 30 plus species. We now are up to 72 on our method, and our UPLC methods are only 6 minutes long. So we've been expanding this. So I'll show you the data, and you'll see again, anthocyanins, phenolic acids, flavanols, and flavanthreols. I'm going to refer mostly to the classes moving forward so we could step through it, but know that we have data on individuals as well. So we tested this out on a mapping population, which actually segregated out for acylated, non-acylated, and acylated polyphenols, and we actually found very good correlation in that. And so then we began the process of looking at, all right, was there differences in content? Yes. Of this, we see high levels of anthocyanins, but a broad range. There's variability where you see the red asterisk is there is significant variation within anthocyanins, within acylated anthocyanins, within phenolic acids, flavanols, and even flavanthreols. There's significant variation across these 30 plus selections from 33 selections from Draper by Jewell in content. Now, bioaccessibility, as you can see, is not related to total content. You see acylated higher than regular, and then you see high levels of flavanols and flavanthreols as well in terms of bioaccessibility, but you see variation there. So our test was to just see, hey, if we have a population that we know is going to be quite variable, can we in fact see this in bioaccessibility as well? So we were able to show it in something as small as 33, and so we decided to scale it up. And so in over 2017, 2018, and 2019, and I'll show you the data, we were able to get now from a diversity panel, 126 successions, 181, and 187. I'm actually going to show you the data from 2017 and 2018, which is what we published. Massimo has the rest of the data now for the 3 crop years, and he is going into that now, and he is actually doing QTL analysis. But I just want to show you the fact that there is that diversity there. So just to give you an idea, flavanols have the highest relative bioaccessibility. So here is a plot: phenolic acids in blue, total anthocyanins in red. So anthocyanins over 2017 and 2018, very similar response in terms of concentration in the fruit. Similarly for flavanthreols and flavanols, 2017, 2018, some difference between crop years, but generally trending the same way. When you look at their relative bioaccessibilities, however, there are some differences year to year. So flavanthreols, you see quite a bit higher in 2018 as a group. Again, same thing for flavanols and phenolic acids, to a modest extent anthocyanins. So again, this immediately would tell us, so it's not just genetics, we also see year by year variation. Is this something that's related to the genetics or not? But overall, what we see is total anthocyanins driving the most, and when we come to absolute content, milligrams per 100 grams, as well as the lower levels of flavanols, but phenolic acids also being an important part of that. When we look at it, I just wanted to point out, since I'm showing it by class, you see a lot of similarities. These are quercetin and centuridin, and then catechin and epicatechin. again, showing that the bioaccessibilities can vary significantly. So when you look within one of these bars, that is the range, the box and whisker plot, and then you see year to year that there could be some differences there. All right, so for anthocyanins, it's actually kind of interesting. In 2017, petunidin and peonidin, which actually are both methylated, just at different spots, quite different. And some of that relates to the content differences in these blueberries. The main ones you see, malvidin, delphinidin, and cyanidin, there are quite a bit different, and cyanidin being quite a bit higher in 2018. What's interesting is consistently what we saw with Draper by Joule, acylated anthocyanins, higher bioaccessibility than non-acylated. And that just means that they were extracted and available for absorption or metabolism by the bacteria. Okay, so it just means they're available and soluble in the gut, but it's consistent with what I think has been reported in that space. And there is variation within each of those. So going from there, try to do some correlations. What we quickly see is that the correlation between content and bioaccessibility is not super strong, which is what we observed in our first screening of 5. The absolute bioaccessibility is very predictive based on content because it's a factor in its calculation. So being a heavy factor of its calculation, it's not surprising it is. But the relative bioaccessibility, in fact, there are a couple of cases where we see some correlation with flavin-triols, with flavanols, but we actually see some negative correlation in terms of some of the anthocyanin species. So it becomes one that is a little bit more complicated. So how do you use these types of data? Again, you can begin to do plots of relative versus absolutes, and we begin to see, for example, some of those very individual genotypes, these PIs, that are actually really outperforming. So these have very high relative bioaccessibility and have very high absolute bioaccessibility as well. So in other words, they are a good combination of content and delivery characteristics. So that's the simplest way to use this. The more interesting way to use this is to then begin to look at the first stage. Let's calculate broad sense heritability and see if there is a genetic basis for bioavailability as a trait, or in this case, bioaccessibility as a trait. Initial calculations on relative and absolute bioaccessibility indicated that there are cases that cross that 40-degree threat, that 40% threshold. So again, looking at the blue and looking at the green here. The pink is content. We kind of saw that already from some of Massimo's original work, that content is going to be tightly tied to genetics. But what we also see here is that relative can also cross that threshold for many of these compounds, not for all. But the point is that there could, in fact, very well be moderate heritability of these traits that relate to this. So where does he go next? The next step of this is looking at the genetics in more detail. So it's suggested that plant genetics might be leveraged in this case. So we're increasing the number that's in. We're done with that now to give him data and enhanced power. He will be doing a QTL analysis now to understand if there are specific genes that might be associated with these effects. That will be where we actually see the benefit back, right? So now we can actually breed back in and see if we can enhance bioavailability in many of these others. So the next other part will be exploring G by P. So there's aspects that we want to do with some of these priority genotypes. And then, of course, the microbial metabolism, as I mentioned, anaerobic fermentation. So we have added that component to our model and are interested in starting that process now. There's a clinical component to this project. Colin Kay, who many of you might know, is actually doing a comparison of a high and low bioavailability genotype that we've identified to actually confirm these data in humans. But again, we can do 1 or 2 at a time there, whereas here now we're able to screen hundreds at a time to at least be somewhat predictive. So I know we're getting ready to run out of time, but I'm going to spend the next 5 to 10 minutes here to switch gears a little bit and talk about my first love, which is carotenoids. So again, something that's quite well tied to the Tufts Center with tremendous carotenoid research out of Tufts over the years. And many of you are very familiar with these. We study extensively the broad spectrum of carotenoids in foods, but we also have over the years expressed a lot of interest in looking at the provitamin A carotenoids as they relate up here, alpha and beta carotene, as they relate to biofortification efforts. In particular, here you see orange maize, which is one that Torbert Rochford's group and Kevin Pixley have been working extensively on. You also see the sweet potato there for sub-Saharan Africa. So we work a lot. I've had the great pleasure of working with Bruce Hamaker's group and others on the Food Process Innovation Lab at Purdue, carried on that work while I was at NC State, working also with the Sorghum and Millet Innovation Lab, almost always looking at strategies for biofortification or food-to-food fortification to enhance delivery of iron, zinc, and vitamin A in staple foods. So we're always interested in understanding, are there factors that can be controlled that drive bioavailability? Because it's also an interesting problem. You can breed a lot, but can you make it available? So this is just an example. We got in early on with the Bill Melinda Gates work on the Africa biofortified sorghum. And so Pioneer at the time, before it was Corteva, Pioneer had developed a transgenic biofortified sorghum. And you can see the color difference. I don't know if it comes out well on your side, but the golden sorghum here versus the wild type high digestibility sorghum. And we were using our models, digestion models, to see, in fact, if we could see if increasing beta-carotene in the sorghum could in fact make it more bioaccessible. So the transgenics were quite good. The null was about 1.2 micrograms per gram. And then here are 3 transgenic events, ABS 168, 188, and 203, where you could see it went from 1.2 to 7.7, 8.3, 5.8. This is great. You quadrupled the amount of beta-carotene. In fact, even more so after preparing it into a porridge, you see ABS 188 is 144 versus 25. So that's a good move. But then after you digest it and you have to have a micellularization event to characterize bioaccessibility, what we found is the transgenic products were quite a bit lower than the null. So at the very end of the day, if you looked at how much beta-carotene was actually being delivered by the transgenic product, there was almost no significant difference between the transgenics and the null, with the exception of the one that was exceptionally high, ABS 188. And at the end, the best you could get after all that gene jockeying was about 15 to 24% of the EAR. Now, in all defense to Pioneer, they did tremendous work, and they've actually increased this number quite a bit more now. We've got some great work that we're working with them on now, but this was actually a little disappointing. And it indicated that you can't just consider total amounts in breeding. You have to think about delivery characteristics. And this is Tristan Lipke's work with myself at Purdue, but it was really a critical component to teach us that we needed to understand the delivery characteristics out of the plants we're working with. So another example, traditional breeding, biofortified cassava from CIAT, and been working at the time with Dominique Dufour when he was at CIAT. And we had 2 graduate students, Ingrid Aragon and Darwin Ortiz, who had been working in my group at the time, that were interested in taking the trip back to Colombia and working in this area. And again, very important, but then you also have to understand the fact that it has to be processed. And cassava is one that undergoes extensive processing, including a fermentation step to eliminate cyanogenic glycosides. So when you think about testing flours or unfermented flours because they're low in cyanogenic glycosides, how is the bioaccessibility of the beta-carotene affected by processing? So just to show you something really quick here, we were interested, of course, on the food science side to make sure we could recover beta-carotene through the process. And this is actually somewhat disappointing. When you start with raw cassava, by the time you get to the end of cooking, in some of these genotypes, you can have as low as 6% of what you started with. So again, something to think about in terms of breeding. We have a whole other program we've worked with there to stabilize compounds through process. But in general, the genetics didn't matter. It was kind of a similar slope. Whether they were high or low, you lost a lot through the process. And fermentation was a key component of that. But what about bioaccessibility? So we took those genotypes and we did them through the in vitro digestion, and we looked at beta-carotene equivalence and the bioaccessibility. And what did we find? Interestingly enough, unfermented in yellow and fermented in red, you have 3 genotypes that if you fermented it, got better. In other words, the bioaccessibility of beta-carotene went up. You had 4 genotypes that did the opposite. If you fermented them, the bioaccessibility went down. And then you had 3 in the middle that seemingly didn't care if you fermented or didn't ferment it. by luck or happenstance, we basically documented a genetic by processing interaction here. And we didn't pick it this way. This was an experiment we added just out of curiosity. But it goes back to tell us that you probably have, depending on the nature of the process you're going to go, to be able to select the genetics for the appropriate process if that is intended to deliver the nutrients. Now, understanding why this is occurring is why we're doing studies sort of like what we're doing with blueberries. A little bit more difficult on cassava, but doesn't mean it can't happen. So interestingly enough, because we had that while we were working on blueberries, we said, what is interesting in the US diet is spinach. Why? Because it's one of the few vegetables that's grown in terms of popularity. And so what if we applied the same principles to the blueberries to spinach? Now the genetics are a little bit more complicated in spinach, but Massimo is a pretty good guy. He's interested in doing everything. So we did the study here with a fairly diverse array of spinach accessions, 71. Now, my background's also in chlorophylls, so this is why you see pheophytin A and B, but I'm not going to talk about chlorophylls today. Let's talk about beta-carotene. And if you look at the diversity of bioaccessibility in 71 spinach accessions, it goes from about 12% to about 45% bioaccessible. Lutein, similar range, but that is broad. That means that there's a lot of differences that might be able to be leveraged through breeding or understanding, in this case, to actually get to enhancing delivery from spinach. Pictured another way, you certainly can see lutein and beta-carotene, both are which critically important, beta-carotene for vitamin A, lutein for eye and brain development, which is quite well known there at Tufts. What can you see? From 69 of those genotypes, you could see, in this case, the content in the bars and the bioaccessible content in the dots. Now, what's interesting is that there are some in here that overperform, that are low in content of the spinach, but actually deliver similarly to the ones that are very high in content, you see? Sorry, I have a very sensitive mouth, I guess. So you're able to kind of begin selection of those for breeding experiments, et cetera. And it gives us an opportunity to enrich it. Like we did with blueberries, look over year to year. We did a fall to spring. There is definitely an environmental impact when it comes to carotenoids, but what's still interesting is while there's a difference, there's a shift in the whole population. You tend to still see the same rankings, i.e. the ones that are high are still high, the ones that are low are still low, but you begin to see a shift season on season, which is actually quite interesting in terms of both content and bioaccessibility. That being said, you know, spinach is consumed in lots of ways. And so this genetics by processing was something we were interested in. And at the time we were doing this, it was really interesting because for many of you that know me, know I worked for Steve Schwartz at Ohio State. Well, Steve started at NC State. So when I first met Steve, he was finishing up a study with Cheryl Rock. And it was really one of the studies that altered the landscape on the carotenoid side, as well as the Ed Giovannucci study at Harvard on lycopene. But when it came to this notion of processed fruits and vegetables being able to deliver more carotenoids, and this study was quite telling because it was a trend for increasing bioavailability of raw carrots and spinach versus processed carrots and spinach. You saw more from the process, but what was interesting is exactly what they fed. So my graduate student comes to me and said, well, this is an old study from Steve Schwartz. And I said, yeah, I know that study well. And she goes, well, they didn't really feed raw product or process. They fed the raw product, but the processed product was actually blended and homogenized. So they basically took canned spinach and canned carrots and put them in a blender and made the participants drink it. That's a different product than something you have to masticate. So he said, well, let's see what happens if we process product. And we look at bioaccessibility to understand. We saw that increase of threefold, let's say, from the Cheryl Rock study was there. So we picked out spinach accessions, which were high performers for us. We did fresh, we did blanched spinach, we did sterilized spinach, so this is canned, and then we did juiced. And I'll really talk about that in a different way. But then we also did homogenates of each of these, and we passed them through. So you have mastication, which we simulated. We actually have a simulated mastication system, which is actually quite good using meat tenderizer, food hammer, and a meat tenderizer, and then a highly homogenized product. Now, why is this important? if some of those differences are driven by the fact that product is different texturally, then you're going to detect those differences also in this model. What we found, again, the general theme is if you process fruits and vegetables, you have enhanced bioavailability. Well, that's actually quite the opposite. What we found across each of the genotypes, from fresh to blanched to sterilized, was they all went down, right? And then juiced was always very high. That was a very different product because you're taking out fiber in this case. What was interesting is looking at what the digesta looked like, and you can really see the masticated product really was not breaking up, whereas the homogenized product was homogeneous. If you plot it a different way, at some point it'll move here. If you plot it a different way, what you actually see here is total carotenoid content in bars, the bioaccessible from the homogenized and the bioaccessible from the masticated. Oral processing is going to be more critical for these compounds than anything else. So if you're not chewing your food well, start chewing it. But what's really interesting is if you actually show the comparisons that were made in the Rock and Schwartz study, it's these two. It's fresh and masticated versus sterilized and homogenized. So now it makes more sense, because our big concern was we're not seeing the effect of processing. Well, we are if we cherry pick the right comparisons. And that's an interesting take-home message in and of itself. So that really was the interesting learning for us there. Now, sorry, I'm having so much problems with this. Another take-home message that we found that was actually interesting was that the processing began to wipe out all the effects of the genetics. So with spinach, unlike cassava, you see a really good distribution, but you really of bioaccessibility, but you don't see it by genotype. You see it clustering by processing type. And what's really interesting is that tells us that maybe we should just be breeding for ones that are higher in this case than anything else, and then match it to the right process type that would actually drive the benefit. So take home messages there, and I'll stop as soon as my computer decides it wants to advance. It's a work in progress, right? It's suggested that plant genetics can be leveraged to enhance delivery of plant bioactives for human nutrition and health benefits. We've been focusing on a lot of the screening, but we're going to start moving more into the genetics by environment and processing, and to be more critical about those, to really understand how to develop plant-based foods, but also recommendations for these. We think that these can be used to leverage breeding programs, but processing strategies as well. So that's the goal for me, is to work with both sides of this, from the farm, also to the processor, but also to the table, to enable people to get more out of the foods they eat. So another thing we're moving on now is we're circling back to where we really started on this. Like I mentioned, some of the work we started on this related primarily to provitamin A biofortification, and working with Davina Rhodes. She recently received the grant to look at provitamin A biofortification strategies, the genetics of this in sorghum. As I mentioned, sorghum was primarily done by transgenic approaches. Part of the reason for that was sorghum was not believed to have enough content or diversity of beta-carotene in the germplasm to warrant starting in traditional breeding. But we did some work early on with Davina to show that, in fact, you definitely see diversity in bioaccessibility. This is over 40 genotypes from a diversity panel, but we're looking already at multiple hundreds now with her, and are excited to actually begin to connect the dots there as well. So I'll stop there and just acknowledge Terry Bolito, who's now at UAMS, started working with her on blueberries at IU, the folks at NC State, in particular, Massimo and Colin Kay, who work very closely with, and of course Purdue, who I still work with, Bruce Hamaker. Actually, Connie Weaver, I should shift her affiliation. She's now at San Diego State. But the great grad students and postdocs who I've had the pleasure of working with, and Michael Zagovich, who's actually now at the Baylor Center for Children's Nutrition. So I had a postdoc working on plants who's now working at Baylor. So slowly but surely, we're all working on the same team. So I'll stop there, and happy to take questions.
Thank you, Dr. Feruzzi, for a very interesting talk, and for providing great insights with regards to the complexity of the pre-ingestive factors in the seed to cell pathway that you described, and its impact on nutrient bioavailability. Our first question is from Dr. Alice Lichtenstein. She asks, you've noted that things like berry skin thickness, solids, and fiber can affect bioaccessibility. Is this variability introduced by the degree of chewing or mechanical disruption? How do fresh compared to desiccated berry preparations compare in terms of bioaccessibility?
So I think that's a really good question. So some of it, as we're learning from spinach, is probably going to be compounded by degree of oral processing. And so there's a lot of variability there. But skin thickness also brings about another point, which is you're also bringing a lot of cell wall material and fermentable fibers and substrates. So some of the things that we've been looking at now, for example, are adding an anaerobic component to this digestion model. We're working with Andrew Nielsen, who has developed a nice model here at NC State that we're hoping to partner with him more and more on while I'm at Arkansas. And so as you put in fermentable substrate, how does that, plus the fact that the microbiota could also alter the phenolic profiles, the combination of both of those substrates being in at the same time, you're bringing fermentable substrates in with the phenolics, you could see very different polar metabolites that are generated. And I think that's how we look at this, is the prebiotic nature. And it's very difficult to use that term because phenolics aren't prebiotics by the official term, but they can be prebiotic-like. And if they're introduced with fermentable substrate, now it's even more interesting. So there's a combination of the variation we get from oral processing. The dried products, as you remove water, are more difficult to chew. You're probably going to be bringing in more intact materials, but you've also disrupted those through the drying process themselves. I think it would be interesting to look at that G by P, if you will, that genetics by processing interaction, to see if there's a relationship there. But I would suspect that there's going to be something if you have variation in skin thickness for sure. That's where a lot of these phenolics also happen to be in blueberry. So.
Great, thank you. Dr. Samin Medani asks if the yearly variability in the level of phenolics and other metabolites, if it's significant enough to impact in vivo effects in animals and humans, particularly in the context of whole diet, where there is a contribution from other foods to the eventual level of these compounds.
Yeah, that's a really great question. So when you look at mixtures and diets, and that's the next step, right? So this is really kind of singly looking at a food and trying to understand the role of the plant genetics, in this case, on a delivery trait. My entire background of research was always looking at food composition, matrix, and interactions from more complex scenarios. So surely they can play a role. And I think that's the next evolution of this, is to try to understand more about the context in which these things are consumed and the variation in response we get. I think some of this relates to what some of you will be working on as you get into the personalized nutrition side of this as well, which is let's stop worrying about individual variation and think about more of it from the population perspective. In this case, I'm looking at the population of blueberries, but as you put that into the more complex diets, I think that's a really great understanding how much of the variability we see in a single food makes itself known in a more complex diet, and then what's the longitudinal change in that over time. The one marker we can look at in our case as phenolics would be phenolic metabolites. So I think that's an interesting experiment to do or think about as you analyze data from broader context of dietary components. And I think that's a really good question because it tells us how complex this really is.
Great, thank you. We have a student who works with Dr. Thomas Biederer and Thomas Biederer, and they ask, historically, was the bioaccessibility of the biofortified foods mentioned, like sorghum and cassava, always low? Or has this decreased from industrialization, just the need for fortification? Also, I may have missed a portion of the talk, but could the same genetic variation of blueberry, for example, have differences in bioaccessibility based on cultivation differences, which you did talk about?
Yeah, so the last one there is absolutely probably will have differences based on cultivation. Okay, so we're getting these from single places. We are doing replicates in some cases looking at that, but these are biological reps from different plants. And if anybody's ever studied phenolics of berries, one side of the plant will be different than the other. So you're definitely going to get a lot of variation based on sun exposure, cultivation, fertilizer, everything for sure. The question is whether or not you get not whether or not you get the variation, but the fact that it's actually going to stay in the same direction, right? But what's interesting on the first question there is thinking about that, like the biofortification program. So yeah, if you think about the fact that have they always been low, you know, some of these cereal grains were bred to have lower and lower amounts of pigments for certain. They've been breeding more for whiter flowers or yellow or light colored flowers for industrialization. But the bioaccessibility component was not one that was really ever known. I don't think we have historic numbers on what the bioavailability out of sorghum was. The issue you get with beta-carotene is historically people will look at numbers that are reported in the literature for cassava and for sorghum and even for millet. And if you look at some food composition databases, they report much higher levels of provitamin A beta-carotene. None of it makes sense. If you actually think about it, those of you that know the carotenoid UV visible spectrum would know that lutein and zeaxanthin, non-provitamin A carotenoids, have the identical spectrum to alpha and beta-carotene. So these older methods were capturing higher levels, but they weren't actually the compounds of interest. My suspicion is we need to know more about how plants capture, store, and then ultimately release these compounds through digestion. In the case of beta-carotene, we know that carotenoids in cereal grains are stored with protein bodies. We also know that in things like sorghum, those protein bodies tend to be less digestible than what you see in their cousin grain maize. And so understanding factors that drive protein digestibility, which is a breedable trait in sorghum. In fact, there are high digestible sorghum varieties to do this in. Those transgenic ones were on the high digestible sorghum varieties. You would be able to better understand if these differences can be brought forward. This isn't an industrialization thing per se, because in many cases, I don't think we've had a lot of beta-carotene in these grains like rice and sorghum and millet probably for hundreds and hundreds, if not thousands of years. So I wouldn't say it's really truly modern industrialization there. It's really trying to understand how we can get more into these staple crops.
Great, thank you. We're past the top of the hour, but we'll take another question or two, which one of which comes from Dr. Shandong Wang. He asks, is it possible that the intestinal microbiota could modulate the food matrix, plant genetics that is, and alter the bioaccessibility and solubility of plant phenolics and carotenoids?
Yeah, that's a really good question. We just got 2 papers published looking at the anaerobic fermentation. And in this case, we looked at grape juice versus whole grapes, really to kind of get at the dietary guidelines thing of 100% juice versus grapes. But we also did it with spinach and the spinach ingredient forms, dried forms, where we looked at the bioaccessibility and intestinal uptake. We added a KCO2 component as well, both in the upper GI and then after anaerobic fermentation. So long story short, I didn't present the data here, but what we find for phenolics is significant release of phenolics in the lower gut by microbial fermentation. And ultimately 100% juice versus whole fruit look about the same in total, but they deliver in different parts of the intestine. You get much stronger acute effects from juice. You get slow and sustained effects from whole fruit. Interestingly enough, a lot of the blood pressure work, a lot of the neurocognitive work on juice on grapes is done with grape juice. It's not done with whole grapes. Similar to spinach, we see very strong stability of carotenoids through the anaerobic fermentation. We don't see metabolism of the carotenoids, but what we do see, and that could very well be because of the fermentation that we're doing, I'm not saying it doesn't occur, but what we do see is ample opportunity for solubilization in the anaerobic compartment as well. And so the question we didn't do in that study is, do any of the genetics of the plant play a role in that? I suspect they could, based on what I was saying earlier. If you have genetics that influence fermentable substrate, I think you will alter what's happening in the lower gut. That's a guess. But yeah, so we just got those 2 papers published. So they'll probably be, I know I just sent the proofs out, but be happy to send those to you to kind of give you some idea of how we've begun screening.
Thank you, Dr. Feruzzi, for that wonderful talk and for sharing all those great insights. And we will definitely keep it in mind for those of us that are involved in post-ingestive factors. We will definitely keep all your great insights in mind. And we thank you very much for sharing your thoughts today.
Yes, thanks. Thank you again for the opportunity.